DNA sequencing by Sanger's chain termination method. Uh, Determining the sequence of nucleotides in a DNA segment or gene is important. And Sanger's chain termination method, because of its ease and simplicity, has found an application in automated sequencing systems. Therefore, in this session, let us look at how Frederick Sanger developed the chain termination method. Let us therefore look at the learning outcomes of this session. DNA sequencing by Sanger's chain termination is based on the synthesis of DNA in vitro, which means effectively replication, using the gene to be sequenced as the template. As the word suggests, chain termination indicates that as the DNA is synthesized in a chain and it is in process, there should be something that should be able to stop the synthesis and therefore chain termination. The termination is possible due to use of a, a special nucleotide, and that is what is called as dideoxyribonucleotide, which is added in the mixture of normal deoxyribonucleotides. Polyacrylamide gel can separate small sized DNA with a difference of just a single nucleotide, and that becomes the essence of this technique. So let us look at the principle or understand the principle and understand the strategies that have been taken by Frederick Sanger to have this method developed. So single-stranded DNA molecules that differ in length by just one nucleotide can be separated using page. So this principle is being used in the chain termination method. So therefore, what is the idea? The idea is that one could resolve DNA fragments of anywhere between 10 base pair to 1500 base pair in a single slab of gel or capillary gel as bands. That means you can actually look at them as bands in a polyacrylamide gel. So you can see over here, this is a band formed because of 10 nucleotides, and this is a band formed because of 50 nucleotides. And between this and this, you have 49 nucleotides long DNA, 48, 47, 46, 45, 44, 43, 42. That way, each of these band is differing from one another by only one nucleotide. And that essentially becomes a very important principle on which this technique is based. The polyacrylamide gel that is used is a denaturing gel that is being used. It's not the native gel, but it is a denaturing gel. Now, the DNA to be sequenced, this is another thing that is important in the method. The DNA to be sequenced should be in multiple copies. That means you should have several copies of this DNA, but the catch is that it has to be single-stranded. So, therefore, the question arises, how do you get single-stranded DNA for sequencing? So Frederick Sanger immediately thought of the use of M13 vectors or phagmids. Phagmids is again M13 plus plasmid, okay, and the phagmid could give rise to single-stranded clones. So therefore, let us consider that in an M13 phage or a phagmid, you have the gene inserted into the vector, and this gene or this fragment of DNA has to be sequenced. So when you want the DNA, this M13 can multiply into several copies, okay, and each of them is a single strand. So effectively what we understand is that this is the DNA that has to be sequenced from here to here. So in order to sequence this, okay, one needs to add a small oligonucleotide which would bind to a region of the M13 vector just before the start of the gene of interest or the gene that has to be sequenced or the DNA that has to be sequenced. Why are we able to synthesize this oligonucleotide? Because we know the M13 sequence. We know the M13 vector sequence, and therefore we exactly know what is the sequence of the M13 here. So when you put the oligonucleotide, it is positioned just at the onset of the sequence that has to be the DNA that has to be sequenced. Now, because we have multiple copies, for every copy you will have the oligonucleotide coming in, binding to that particular region where it finds its complementarity. So if you look at it in a resolved form, this is the sequence. From here, this is the sequence that has to be, this is the DNA which has to be sequenced. And this over here is the oligonucleotide that has attached itself to the M13 phage or M13 vector. So this oligonucleotide in fact acts as a primer, and so you have a free 3' OH group that is present, and this free 3' OH can be used by the DNA polymerase to synthesize the DNA, which is what is the second principle of the Sanger's chain termination method, that is, replication of a single strand should be possible. And so this sequence that has this DNA that has to be sequenced is present in the form of single strand, and just upstream of that is attached a primer, and using this primer this DNA segment can be synthesized. So that is the second principle on which you have the chain termination method based. Which means that if this has to be synthesized, what is needed definitely is DNA polymerase. Now you cannot just use any DNA polymerase. The DNA polymerase that has to be synthesized, that has to be used in the chain termination method should be generally devoid of other enzymatic activities, which means we understand that DNA polymerase can have exonuclease activities. If you take DNA pol 1, it can have 3' 5' exonuclease activity as well as 5' 3' exonuclease activity. But what is required in the chain termination method is only the polymerase activity. And therefore, one has to modify the DNA polymerase in such a way that it does not have its 3' 5' or 5' 3' exonuclease activity existing. So, Lino fragment can be used. Lino fragments are generally DNA polymerases devoid of its exonuclease activity, or what can be used is specialized DNA polymerases like sequenase that is obtained from T7 bacteriophage, and that sequenase also can synthesize the DNA without the 3' 5' exonuclease activity. Now, in a while, we will understand why we do not need the exonuclease activity. But here, what is important to notice that the DNA polymerase that you use cannot be just any DNA polymerase. It should not have the other 3' 5' exonuclease activity in its capacity. It should only have polymerase as its capacity to carry out synthesis. What is of course needed is the DNTPs. So all the DNTPs, whether it is DATP, DGTP, DCTP and DGTP are, sorry, this will be DTTP, are required for synthesis of the chain. Now the question therefore that arises is, we have already looked at how the chain is going to be synthesized. But the word termination suggests that one has to stop the synthesis of the chain. So the question that comes up is, how can one terminate the chain being synthesized? So Frederick Sanger used a very simple strategy. And the strategy was that a DNA polymerase is able to synthesize DNA only when it gets a 3' OH group, and that 3' OH is an existing DNA or it is present in the primer. So DNA polymerase is able to elongate a DNA only when it has 3' OH. So therefore, what if the 3' OH is not made available? If 3' OH is not made available, then DNA polymerase can no longer continue to synthesize the DNA. So how can that be done? So what was done is that dideoxyribonucleotides were used by Frederick Sanger and the team. Now, what is dideoxyribonucleotide? So, generally, when you look at the deoxyribonucleotides, the sugar, so it has a base with a glycosidic bond to the ribose, deoxyribose. When you say deoxyribose, the 2' carbon does not have an OH group, so therefore deoxy. And then the 5th carbon of the sugar is attached to the phosphate moieties. In case of dideoxyribonucleotides, the third carbon, the 3' carbon, also is devoid of an OH group, so it only has an H. So therefore, this specialized nucleotide does not have an OH at the 2' carbon, nor does it have an OH at the 3' carbon. And therefore, it is called as dideoxy, because from both the carbons you have 2 oxygens removed. Now, effectively, what does it mean? It means that the DNA polymerase can incorporate this nucleotide, but because there is no 3' OH, now it cannot further add any more nucleotides to this nucleotide. So DDNTP is therefore important. So for example, you have the DNA, single-stranded DNA, which is used as a template. So this is the DNA that has to be sequenced. This is the primer that is being attached. And this primer has a 3' OH end over here. So you have a deoxyribonucleotide being added, another deoxyribonucleotide being added, another added, and another added, and another added. So it can continue to add deoxyribonucleotides, as it means the DNA polymerase can continue to add the deoxyribonucleotides as long as a 3' OH group is available. So this will continue until you have a dideoxyribonucleotide added. Now, when you have a dideoxyribonucleotide added, it has been added because there was a 3' OH previous. Over here there's a 3' OH, so therefore this can definitely be added. But now the dideoxyribonucleotide does not have a 3' OH, nor does it have a 2' OH. So the DNA polymerase will not be able to add the next nucleotide, and hence synthesis of DNA will terminate at this point. So the catch is that there is no 3' OH when a dideoxyribonucleotide is added. So DNA polymerase, this is the fact that has been used to design or to develop this method. DNA polymerase needs a nucleophile to synthesize further, and when you deprive DNA polymerase of the nucleophile, then it cannot synthesize any further. It cannot elongate the chain any further, and therefore there will be termination. So one thing that can be asked is, if the chain terminates and if you have a high number of DDNTP, okay, then because you have a high number of dideoxynucleotide triphosphates, the first nucleotide that is attached may be the dideoxynucleotide triphosphate. So you may not have further chain elongation at all. Now, if the situation that you have a higher amount of DDNTP in comparison to DNTPs, then the first nucleotide will always be dideoxyribonucleotide triphosphate. So therefore, the strategy that is used is that the ratio of DDNTP to the DNTPs has to be maintained, which means that the levels of DNTPs have to be much higher than the level of DDNTP. The concentration of DNTPs have to be much higher than the concentration of DDNTPs. Only if that is the case, you can have chain elongating to a certain extent. Sometimes the entire chain will be formed but no DDNTP is added, because it depends on how much amount of DDNTP is available to the DNA polymerase to add. So therefore, looking at this condition, let us consider reaction mixture 1. In reaction mixture 1, you have all the DNTPs added, DNA polymerase added, the template added, the primer added, and DDATP. So a very specific dideoxyribonucleotide is added to reaction mixture 1. What does it indicate? Say this is the T. So against T you will have an A being added. Now suppose at this point dideoxy ATP is added, then the fragment length, that is the chain that is synthesized, will be this long. Suppose you have the DDATP not added over here but it is added over here, then the chain length will be this. Suppose the DDATP is neither added over here nor added over here but is added over here, then the chain length is this. What does it indicate? It indicates that as many times the and plus you must understand the reaction mix has not just one template, it has several templates. Correct? We have already seen that in one reaction mix you will have several templates present. You'll have several DNA polymerases, a lot of NTPs, and a specific concentration of DDATP. That too very specifically only one DDATP. So you can have no DDATP added until here. You can have no DDATP added until here or here. So the outcome is that based on where the DDATP is added, you will have different sized fragments formed. So in this reaction mix, after the reaction is complete, what you will get is n number of different sized fragments. You will not have just one sized fragment being formed, but three different sized fragments being formed. So that is very important. And each of the fragment is going to end with the A, and this A is a dideoxyribonucleotide. So the ratio, I'm repeating, the ratio of DNTPs to DDATPs is therefore highly important. If you have a high amount of DDATP, then you would always have the chain synthesized till here, and here the synthesis will stop. Why? Because the amount of DDATP is high, it will always add a DDATP. But because the amount of DATPs is higher, you may not have a DDATP being added over here. It may be added just about here. Okay, so based on that, you would have different sized fragments being formed. Now, likewise, you can have reaction mix 2. In reaction mix 2, everything is added, but the DDATP, the dideoxyribonucleotide that is added is DDCTP. So in this case, again, suppose here you have a G, here you have a G, and here you have a G. So the dideoxy-CTP can be added over here, can be added over here, and can be added over here. So if it is added over here, the chain elongates only this much. If it is added over here, the chain elongates this much. If it is added over here, the chain elongates this much. So again, what is the outcome? That you have different sized fragments obtained ending with C. Likewise, one can carry out reaction mix 3, that is specifically for DDGTP. You have reaction mix 4, that is specially carried out with DDTTP. So you will get an A family here, you will get a C family here, you'll get a G family here, and you will get a T family here. And each one would have different size fragments ending with the corresponding dideoxyribonucleotide. Okay, so this is how chain is synthesized and terminated. Now let us look at what we should do further. Okay, important to be noted is that that all the four DDNTPs that are used can be labeled with a fluorescent tag, because then you can identify, okay, whether it is A family, whether it is C family, whether it is T family, or whether it is G family. So the fluorescence is going to tell whether it is an A family or whether it is a G family likewise. Now, how does one decipher the sequence? Okay, so the reactions have been carried out. Now is the time to actually analyze these different fragments and find out the sequence. So how is it done? So therefore, for every fragment, if one checks the dideoxyribonucleotide, then you kind of get an answer. Now, suppose you run the gel. So this would have the reaction mix where the dideoxy ATP was used. This is the reaction mix where dideoxy CTP is used. This is the reaction mix where dideoxy GTP was used. And this is the reaction mix which had dideoxy TTP. So this is A family, this is C family, this is G family, and this is the T family. And like we said, you would get different fragments. Okay, and these fragments would be separated out in the polyacrylamide gel electrophoresis, depending on, or even agarose gel electrophoresis for that matter, depending on the size of the fragment. So we all know that in agarose gel electrophoresis, The smallest fragment or the smallest DNA molecule will move the farthest from the well, and that one which is the longest will move the least from the well. So you have all these bands separated based on their molecular size. So say for example, this is the DNA that has to be sequenced, that is the template DNA, this is the primer. So let's say for example, this is G. This is okay. So when you have synthesis happening from here, okay, and if the chain terminates at this point, okay, then this would be the smallest nucleotide or the smallest DNA molecule that is possible, right? So let's consider that in the C family, of all these fragments, the C family has moved the farthest. So if this is C, then complementary to that. So we know that what has been added is C, okay, and this belongs to C family. So what is therefore complementary to C is G. Then the next one is T, okay. So what is complementary is A. What is the next one? It is T. So what is complementary is A. Then you have the G. Then you have the C. Then you have the A. Again an A. Then you have a G. Then you have a A. You have C. You have T. You have C. You have G. You have T. You have A. Correct? So when we said A over here, the complementary is going to be T. When we say T over here, the complementary is going to be A. So the template sequence is this, because what you read in the gel is its complementary. Okay, so I'm repeating again, depending on how long the fragment has been synthesized, okay, you have the smallest fragment, and the smallest fragment is C. So you say put a C over here, then you put a over here, sorry, a T over here, a T over here, a G over here. So if you keep writing the sequence based on what is present over here, its complementary will be this. So this is the sequence that that one has been trying to analyze. Now, when one does an automated system like mentioned, each of the dideoxyribonucleotide can be attached to a fluorescent tag. So, for example, if A is orange, T is blue, G is green, and G is red, and these fluorescent molecules are detected in an imaging system, then one can see the peaks that are formed, and based on the peaks that are formed, you can actually kind of look at when you analyze this gel, you can look at the sequence, and from this you will be able to see the sequence, because the peaks are going to tell which dideoxynucleotide is present, and based on that you can get a sequence in the automated sequencer. So this is how chain termination method has enabled one to decipher the sequence of the DNA. So let us make the conclusions. In Sanger's chain termination method, that is based on the principle of replication, a template is needed, DNTPs are needed, and DNA polymerase is needed. To terminate the synthesis, dideoxyribonucleotides are used in the reaction mixture, which can be incorporated by the DNA polymerase, but from that point on, elongation stops as there is no 3' OH end available. Post the reactions, the different size fragments are separated in a denaturing polyacrylamide gel, and then these fragments can be identified by the dideoxynucleotide end and deciphered to find its complementary sequence. Therefore, Frederick Sanger's chain termination method for DNA sequencing has been adopted for several fast-paced sequencing methods today. It is important to note how simple strategies can help develop a very important method. Thank you.